Oh, thank you, Tegi.
Thank you, Tegi.
Hi guys. We want to ask about transcription.
And I want to ask you about translation. Can you explain this to us?
Huh? You don't understand that? It's just a simple topic, right? You just should study it yourself. I don't have time to do that.
Oh, I'm sorry for this to be you, Huda.
Never mind, we'll just find another person.
Maju. Maju.
How stingy. Guys, you should read this.
Allah yang bertakwa, iman tak dapat diwarisi dari seorang ayah yang bertakwa.
Transcription and translation are two of the most fundamental biological processes that allow cells to decode genetic information and produce the proteins necessary for life. So, what is transcription? So, transcription is the process by which genetic information stored in DNA is copied into complementary molecule called RNA. This RNA molecule then serves as a template for synthesis of proteins, the functional workhorses of the cells. And what is translation? So translation is the process where mRNA is translated into a sequence of amino acids. Transcription occurs in nucleus. So what does involves in transcription process? So first of all, we got DNA. So DNA is the genetic blueprint that stores hereditary information. And then we got RNA polymerase. After that, we got promoter. So promoters is DNA sequences that provide a binding site for RNA polymerase to initiate transcription. Last but not least, we got terminator. Apart from that, there are also stages of transcription. So the first stage of transcription is initiation. So initiation is where RNA polymerase binds to the promoter region of a gene and unwinds the DNA double helix. And the second stage of transcription is elongation. Elongation is where RNA polymerase moves along the DNA template, synthesizing a complementary RNA strand nucleotide by nucleotide. And the last stage of transcription is termination. Termination is where RNA polymerase encounters a termination signal and releases the newly synthesized RNA molecule.
Initiation. Commonly, it includes a data box about 25 nucleotides upstream from the transcriptional start point. Several transcription factors, one recognizing the data box must bind to the DNA before RNA polymerase II can bind in the correct position and orientation. Additional transcription factors bind to the DNA along with RNA polymerase II, forming the transcription initiation complex. RNA polymerase II then unwind the DNA double helix and RNA synthesis begin at the start point on the template string. Elongation. As RNA polymerase II moves along the DNA, it untwists the double helix, exposing about 10 to 20 DNA nucleotides at a time for pairing with RNA nucleotides. RNA polymerase II adds free RNA nucleotide to the 3' end of the growing mRNA molecule as it continues along the double helix. mRNA is synthesized from 5' to 3' direction. As transcription process forward, the newly synthesized mRNA molecule behind the RNA polymerase II peels away from its DNA template and the DNA double helix reforms.
To the final stage of transcription, which is the termination, where the RNA polymerase stops transcribing and releases the newly formed RNA strand. Transcription continues until the RNA polymerase reaches the terminated sequence on the DNA, which signals the end of transcription. In eukaryotes, RNA polymerase II adds nucleotides to the mRNA until it reaches the polyadenylation signal sequence, which causes it to detach from the DNA template and release the completed mRNA. Before moving on to the translation, RNA splicing process occurs in eukaryotes where the mRNA is modified before leaving the nucleus. Introns, the non-coding sequences are removed and exons coding sequences are joined together to form the final mRNA. A 5' cap is added at the RNA's 5' end and a poly A tail is added at the 3' end to help stabilize and guide the mRNA. So, we are moving on to the translation process where the genetic code in mRNA is decoded to form a sequence of amino acids to create proteins. This process occurs in cytoplasm. The main enzymes involved are amino acyl tRNA synthetase and peptidyl transferase. Translation requires three key components, which is mRNA, tRNA, and the ribosome.
Next is initiation. A small ribosomal subunit binds to mRNA at mRNA binding site. After small ribosomal subunit bind, and initiate the tRNA with anticodon 3' UAC 5' base paired with start codon 5' AUG 3'. The initiated tRNA carries amino acid methionine. Next, large ribosomal subunit bind and complete the translation initiation complex. The initiated tRNA is in the P site while the empty A site is available for next tRNA. The next process is elongation, which is codon recognition. An aminosyl tRNA with anticodon bind with mRNA codon in A site by complementary base pairing. mRNA codon in A site form hydrogen bond with the anticodon of an incoming molecule of tRNA carrying its appropriate amino acid. Codon recognition requires hydrolysis of one molecule of GTP.
Peptide bond formation. rRNA catalyzes the formation of peptide bond between the carboxyl group of growing polypeptide on tRNA in the P site with amino group of new amino acid on tRNA in the A site. This step separates the tRNA at the P site from the growing polypeptide chain, and the chain is transferred to the tRNA at the A site. Translocation. Ribosome moves to the next codon of mRNA, which is moved along mRNA template from 5' to 3'. The tRNA with polypeptide chain from A site is translocated and moved to P site. The empty tRNA in P site is translocated and moved to E site. Then it is released from ribosome. The A site is available for next tRNA. Translocation ensures that mRNA is read from 5' to 3', codon by codon. The cycle is repeated until a complete polypeptide chain is formed.
Termination. Elongation continues until ribosome reaches termination codon, stop codon UAA, UAG, UGA on mRNA in A site. A protein called release factor binds to stop codon in A site and hydrolyzes the bond between polypeptide and tRNA in P site. This frees the polypeptide and the translation complex assembly dissociate with the hydrolysis of two GTP molecules.